Good morning and a very warm welcome. I am Anchit Dikshit from MBBS Batch 2024 from ASGSA TDS Medical College, Fatehpur, Uttar Pradesh. And in this presentation, I am going to talk about classification of carbohydrates. We'll first take a look at what carbohydrates are. We'll then move on to their classification systems. We'll take a look at each of these broad categories and then their biomedical importance. Let us first define carbohydrates. You must have heard about glucose, fructose, lactose, cellulose, starch, chitin, and inulin, such polymers in your prior classes. Now, if you define it in a layman language, they can be defined as carbo, which means that compounds that have carbon, and hydrates, which means they have H2O or water, giving them the general formula CnH2O whole N. The problem with this definition is that if you follow this definition, a compound acetic acid, which is not a carbohydrate, will be included in this category. So how do we tackle that problem? We can do that by defining carbohydrates in a better manner so that these compounds are entirely excluded. Now, we define carbohydrates in biochemistry as polyhydroxyaldehydes or ketones or compounds which give polyhydroxyaldehydes or ketones on their hydrolysis. This excludes compounds like acetic acid entirely from the classification. Now, these are some common carbohydrates and their classification system, which is broadly like this. We see that on the basis of number of their constituent units, carbohydrates can be classified into monosaccharides, which do not give any simpler carbohydrate on hydrolysis, oligosaccharides, which give 2 to 10 monosaccharide units on hydrolysis, and polysaccharides, which give more than 10 sugar units on their hydrolysis. Each of these are further classified into various categories, and we'll take a look at them further down in the presentation. Moving on to monosaccharides. These are sugars that cannot be hydrolyzed further to give any simpler carbohydrates. They have the empirical formula CnH2O whole N, and they show a peculiar phenomenon which is called ring cyclization if they have 5 or more carbon atoms in their chain. Now their further classification can be done on the basis of number of carbon atoms or on the basis of functional groups. Let us see how. If we classify them on the basis of functional group, they can be divided into aldoses, which have aldehyde group as a functional group, or ketoses, which have a ketone group as a functional group. If we classify them on the basis of number of carbon atoms in their carbon chain, they can be trioses, tetroses, pentoses, hexoses, heptoses, and so on. Now, if we combine these two classification systems, something very interesting happens. If we look at the columns, we see that there's an aldose, and if we take a look at the rows, there's a triose. So aldotriose sugar, for example, we have glyceraldehyde at that position. So glyceraldehyde is an aldose sugar which has 3 carbon atoms. Therefore, it can be called an aldotriose sugar. Similarly, fructose, which is a common example given in carbohydrates, is a ketohexose sugar because it contains ketone group and has 6 carbon atoms. Now, to join the monosaccharides into a larger structure like a disaccharide or a polysaccharide, we need a type of bond which can achieve that function. That function is performed by the glycosidic bond. This glycosidic bond is formed by condensation, that is, elimination of water from the both sugar molecules combined. Now there can be different types of glycosidic linkages as we will see further in the presentation. Here I have attached an example of formation of sucrose from glucose and fructose. Now these are disaccharides and they yield 2 monosaccharides on hydrolysis. We have taken a few examples like maltose, lactose and sucrose. They are made up of 2 glucose units, galactose and glucose unit, glucose and fructose respectively. Now something to note here is that each one has a different kind of linkage like alpha 1,4 linkage in maltose, beta 1,4 linkage in lactose and an alpha 1 beta 2 linkage in sucrose, each formed from different kinds of sugars. Please do note that alpha 1 beta 2 linkage of sucrose renders it unable to utilize its anomeric carbon in reduction and oxidation processes, which makes it a non-reducing sugar. Now moving on to oligosaccharides. These have 3 to 10 monomeric units. Now, while some would consider disaccharides to be within oligosaccharides, I've taken them separately. Now, while some would consider disaccharides to be part of oligosaccharides, I have included them separately. Oligosaccharides give 3 to 10 monomeric units on hydrolysis. A few examples would be maltotriose, which is made up of 3 glucose units, raffinose, which is made up of fructose, galactose, and glucose. Each one is a different sugar. Stachyose, which is formed from 2 galactose molecules, a glucose molecule, and a fructose molecule. Verbacose, which is a pentasaccharide formed from 3 galactose molecules and a glucose and a fructose molecules combined with the help of that glycosidic bond. Polysaccharides are the compounds which yield more than 10 monomeric units of monosaccharides on hydrolysis. Now, these can be of two types: homopolysaccharides, which have the same type of monosaccharide in the repeating unit, or heteropolysaccharides, which give more than one unit in their repeating chain. Homopolysaccharides, for example, have only one kind of monomeric units and would include compounds like starch, glycogen, cellulose, inulin, chitin, and dextrin. Please do note that starch, glycogen, cellulose, and even dextrin are polymers of glucose, but they are different compounds in the sense that their linkage is different, which makes their chemical properties vastly different. Now we have to note here that starch and glycogen have both a similar kind of linkage. Even then they are different compounds because they differ in their branching structure, where glycogen is more heavily branched. Compounds which are heteropolysaccharides give two distinct monosaccharide units on hydrolysis. For example, agar is a heteropolysaccharide made up of galactose sulfate and glucose. Heparan sulfate, which is made up of T-glucosamine and D-glucuronate or L-iduronate. Here I've included a table from the book U. Satyanarayan. These include common heteropolysaccharides, their composition, and the functions they perform within the body. Let us take a look at the biomedical importance of these categories of carbohydrates. Monosaccharides are an important class in which glucose is included. It is an important sugar that provides energy for various metabolic reactions and mechanical functions performed within the body. It is a major source of energy for every task that we perform in our daily lives. It is so important that it is called sugar fuel of life. It is an important intermediate in glycolysis pathway, and it is the monomeric unit of various important polysaccharides as we have seen in a previous slide. Glyceraldehyde is a triose sugar that is involved in glycolysis. It is an intermediate within the pathway. Ribose is present in RNA and DNA as 2'-deoxyribose. Erythrose, ribulose, xylulose, and pseudoactylose are intermediates of HMP shunt pathway. Fructose is present in fruits and honey, and it is converted to glucose in liver. Mannose is a constituent of glycoproteins and glycolipids and in blood group substances. Among disaccharides, major ones like sucrose, lactose, and trehalose hold immense importance. Sucrose, for example, is commonly implicated in dental caries. If we consume too much sugar, our teeth will rot by the production of acid that the bacteria present in our mouth produce and which degrades the enamel of tooth and slowly the tooth decays. Lactose is present in milk and dairy products, and you must have heard about lactose intolerance, which is a disease which renders one unable to digest lactose, and it can also express itself in the form of lactose intolerance symptoms like an anaphylaxis reaction. Trehalose prevents protein misfolding, so it is a protective sugar present within the body. Oligosaccharides function as blood group antigens. They form glycoproteins and form glycolipids. They are present within the cell membranes of various cells within the body. Homopolysaccharides, for example, cellulose, starch, glycogen, inulin, dextrin, and dextrin, hold various important positions within the biological plane. For example, cellulose is a dietary fiber. It is indigestible by humans, but even then it is important in our diets because it prevents constipation, colon cancer, and diverticulosis by providing bulk and volume so that the food keeps moving. Starch is the main dietary source of glucose for humans because humans cannot synthesize their food directly, and starch is the form of carbohydrate that is synthesized by plants, and we obtain energy from it through plants. Glycogen is stored in liver and muscles, and it is involved in the maintenance of blood glucose homeostasis. It is the stored form of energy present within animals. Inulin is a polymer that cannot be digested by humans, but it is important in lab diagnostics like inulin clearance test in the determination of GFR, which is glomerular filtration rate, a kidney function test. Dextrans are used intravenously as plasma volume expanders for treatment of hypovolemic shock. Dextrans are high molecular weight carbohydrates which are produced by bacteria, so they do not go out of circulation really easily. And since they are osmotically active, they'll provide fluid volume so that the patient can recover from hypovolemia. Dextrins are partially hydrolyzed products of starch and hence they are easy to digest and included in baby foods. They are also used as mucilage and pastes. Among heteropolysaccharides, important ones like agar are used as culture medium in laboratories for growing bacteria and fungi and microorganisms. It is also used occasionally in constipation treatment as a laxative and a supporting matrix in gel electrophoresis, which is a diagnostic technique used in laboratories. Dermatan sulfate plays a structural role in sclera and maintains the shape of the eyeball. Keratin sulfate is present in cornea and plays a role in corneal transparency. Hyaluronic acid occurs in synovial fluid present between the joints, vitreous humor of the eye, and umbilical cord and cartilage. It provides a cushioning and lubricating effect along with preventing infections. Heparin is an anticoagulant synthesized by mast cells. Heparin sulfate is present in glomerular basement membrane and it helps it maintain selective permeability, which is important for renal function. There are a few derivatives of monosaccharides like uronic acids, which are obtained by oxidation of monosaccharides at the terminal hydroxy group, sugar alcohols, which are formed by the reduction of carbonyl group, sugar phosphates, which are esters of hydroxyl groups of monosaccharides with phosphates, amino sugars, which are formed by substitution of hydroxyl groups of monosaccharides with an amino group. Now, to end the presentation, I would like to say that to classify something, we must know about it so that our broad knowledge can be summed up in a systematic manner. And to understand something, we have to collect that knowledge in the form of a classification system so that we can further deepen our understanding of such compounds. Thank you.